To understand life, evolution, and our place in the universe, we shall start at the beginning. Origin of life research is one of the most challenging fields of biology. The transition from lifeless matter to a living cell is still a persistent enigma. Fossil evidence seems to suggest that it happened relatively soon after the late heavy bombardment, roughly 4 billion years ago. But we can't observe this transition. We can only speculate. According to a leading researcher in the field, Cronin, biology is just chemistry with history.
All that biology is chemistry with history.
Is that so? Although we cannot observe the transition from lifeless matter to living beings, we can observe the transition between life and death. So, what differentiates a living cell from one that just died? Is it chemistry with history? Is it information? Is it repeating patterns? Not really. It's actually function. If the end of life is the loss of function, then to start life, matter has to gain function. Functions such as metabolism, homeostasis, and reproduction. However, in a cell, all of these processes can be traced back to gene expression. Even if wounded or cut in half, as long as gene expression works, the cell is technically alive. The death of a cell, the point of no return, is reached when gene expression, the program code of life, terminates. So it seems to be the case that in order to get a cell, one needs to give matter a functioning program code. Let's see why by understanding gene expression and protein synthesis. There is no easy way to carry out the basic functions of life. Even in the simplest bacteria, these processes need clockwork-like mechanical molecular machinery. The machines we are talking about are unique macromolecules called proteins.
And the case I want to make for you is that proteins are machines. You have 20,000 odd different types of machines in your body, and then other kinds of living organisms have other kinds of protein machines. These are real machines. That's not a metaphor. They use energy, they spin around, they pump, they act to cause force and motion. And a critical point that I want to make about how proteins function in general is they function by shape, and by shape changes. And you could see that protein molecule going back and forth as different chemicals come and go.
Proteins are built by chaining together hundreds or even thousands of amino acids. Although you can chain together any odd sequence of amino acids, the order bears great significance. Different amino acids have different side chains. The complex interactions between these side chains determine the shape and function of the resulting protein. You can think of these side chains as magnets, springs, and joints. When they properly align, they drive the folding of the linear amino acid chain into a stable three-dimensional structure. But finding such an alignment is not easy, and that's an understatement. The overwhelming majority of possible amino acid chains don't fold into a functioning protein. It's estimated that the chance of finding a functioning sequence could be as low as 1 in 10 to the 77th power. Let's say I mark an atom in some way, then let it loose in the galaxy. If we consider the estimated number of atoms in the Milky Way galaxy, and we randomly pick just one atom, the likelihood of picking the correct atom on the first try would be much higher than randomly generating a functioning protein sequence. However, most biologists don't consider these odds to be a significant obstacle for the evolution of life. According to British evolutionary biologist Richard Dawkins,
it is amazing that you can still read calculations like the hemoglobin calculation used as though they constituted arguments against Darwin's theory. The people who do this, often expert in their own field, astronomy or whatever it may be, seem sincerely to believe that Darwinism explains living organization in terms of chance, single-step selection, alone. Chance is a minor ingredient in the Darwinian recipe, but the most important ingredient is cumulative selection, which is quintessentially non-random.
However, when we are talking about the origins of the first proteins, cumulative selection offers little to no help at all. But let's consider his reasoning.
I don't know who it was first pointed out that, given enough time, a monkey bashing away at random on a typewriter could produce all the works of Shakespeare. Let us limit the task facing our monkey somewhat. Suppose that he has to produce not the complete works of Shakespeare, but just the short sentence, "Methinks it is like a weasel." It isn't difficult to calculate how long we should reasonably expect to wait for the random computer, or baby or monkey, to type, "Methinks it is like a weasel." So much for single-step selection of random variation. What about cumulative selection? How much more effective should this be? Very, very much more effective. We again use our computer monkey, but with a crucial difference in its program. It begins by choosing a random sequence of 28 letters just as before. The computer examines the mutant nonsense phrases, the progeny of the original phrase, and chooses the one which, however slightly, most resembles the target phrase, "Methinks it is like a weasel." This goes on generation after generation. After 20 generations, the phrase chosen for breeding was. By 30 generations, there can be no doubt. Generation 40 takes us to within one letter of the target, and the target was finally reached in generation 43.
Unfortunately, this analogy is fundamentally flawed. In this program, a sentence which matches the target phrase by 40% is selected over one that matches it only by 20%. But that's not how proteins work. An amino acid chain that matches a protein sequence by only 40% does not fold into a functional shape. As a result, it does not serve any purpose for the cell and is equally useless as any other random sequence. Therefore, there is no selective pressure to preserve such a chain. In other words, it won't be selected. So there is no cumulative selection. Once you have a library of working proteins, it's feasible to get variations by slight modifications. But we have the same problem if, in order to transition from one functioning protein to another, it must go through successive, non-folding steps. To consider this condition, that a protein must fold into a functional shape, this is how we can modify the monkey program to make it truly reflect the challenge evolution is facing with completely novel proteins. Let's transform the phrase, "Me thinks it is like a weasel," to "Your behavior hath struck her into amazement and admiration," with the constraint that with each step we must get a meaningful English sentence. It's an impossible task, even for a supercomputer. Now that we have established that assembling proteins randomly is not feasible, let's see how living cells actually do it. And this is where DNA enters the picture. All the sequence-specific information required to build proteins can be encoded in DNA. What makes this process particularly fascinating is that DNA itself is not physically or chemically related to amino acids or proteins. Instead, the relationship between DNA and proteins is purely semantic, where DNA is, essentially, a digital data storage medium.
I could fit all movies ever made inside of this tube. So anything, really anything, that can be stored as zeros and ones can be stored in DNA. We just have to convert our data, all of those zeros and ones, to A's, T's, C's, and G's.
It's not just us. The cell also uses its DNA as a versatile data storage medium. The vast majority of DNA doesn't code proteins. Unfortunately, for decades, these non-protein coding parts were viewed as junk by the scientific community. But more in-depth research revealed that this couldn't be further from the truth. These parts play crucial roles in gene regulation, error correction, RNA regulation, epigenetic regulation, and chromosomal stability, just to mention a few. When it comes to encoding amino acid sequences, it's merely an issue of informatics. The task at hand can be phrased like this. How can you represent 20 different kinds of amino acids by using only 4 different characters? Self-evidently, using a single nucleotide would only allow for 4 possible amino acids to be encoded. Using 2 nucleotides would allow for 16 possible amino acids, which is still not enough to represent all 20. Therefore, a minimum of 3 nucleotides is required to represent all 20 amino acids. But 3 nucleotides allow 64 possible 3-letter combinations, so there is wiggle room for using multiple codes for every kind of amino acid. This wiggle room also means that there is an astronomical number of possible arrangements. There is essentially an infinite number of ways to create different codon assignments. From this almost infinite sea of possibilities, the one arrangement chosen for life is very special. This is the universal genetic code of life. This particular arrangement is optimized to the absolute extreme to prevent random mutations having an effect. It is fine-tuned to preserve the initial function of proteins and to prevent change through mutations. It's also optimized around the amino acid needs for protein stability. Considering these aspects, it's estimated that our code is optimized beyond 1 in a billion. We know that this genetic code is the same since Luca, the last common universal ancestor. But to explain its special features, it's theorized that there was a preceding era while it was being heavily optimized. However, this leads to the paradoxical view that this system was optimized by the effects of blind mutations to prevent the effects of blind mutations. Let alone the self-defeating nature of this proposed process, optimizing the genetic code with purely Darwinian mechanisms is technically unworkable. Much like how changing the encoding of a document breaks the text it contains, changing the genetic code would break the genome of the organism. What you see here is an abstraction. This table, in this form, does not physically exist within your cells. Instead, there are molecular agents that are responsible for knowing and applying the rules. Codons are merely symbols. You can place a piece of messenger RNA with a perfect mixture of all 20 types of amino acids into a petri dish and add fully functional ribosomes, but you won't see any proteins forming, no matter how long you wait. This is because symbols alone don't achieve anything. You can give a monkey a map to a lush banana grove. It won't be any help for him. Symbols are essentially meaningless without a system that connects them to their intended meanings. In living cells, there are unique agents known as amino-acid tRNA synthetases that play this role. These molecules serve as the keys for the genetic code, with each type of amino acid having a corresponding tRNA synthetase. However, the task of connecting symbols to their meanings is far from trivial. One end of the molecule must read and recognize codons, while the other end must recognize and attach the corresponding amino acid. What we have here is truly remarkable. We have a molecule that utilizes symbolic representations to reference other molecules, DNA, and an information processing system that comprehends these symbols and links them with their corresponding meanings. The emergence of such symbolic and language-like features in physical matter presents a conundrum that still eludes explanation.
Translation occurs in structures called ribosomes. So the messenger RNA is read off the DNA, and in the ribosome it is pulled through like a tape through a tape recorder, and the sequence of those nucleotides specifies the sequence of amino acids. So this building chain of amino acids then forms the protein. Another thing that we can notice about this is that there are enzymes or proteins involved in each of these processes. So none of this, although there are RNA molecules involved, none of this can happen without enzymes, without proteins. So that leads to a kind of chicken and egg problem, right? Where you have genes that store information, that encode proteins that make enzymes, and you need those enzymes to catalyze the reactions to copy the genes. So how do you get out of that problem? The RNA world is the way you get out of that problem.
The idea here is that at one point the chicken was the egg. Both the chicken and the egg were RNA. The RNA world describes a hypothetical era before proteins and DNA in which all the roles these play in modern organisms were believed to have been carried out by RNA molecules.
And Francis Crick the next year said that possibly the first enzyme was an RNA molecule with replicase property. Thus, a system based mainly on RNA is not impossible.
There are a couple of problems with this approach. More than 50 years passed since this idea came about. The understanding of molecular biology has undergone enormous progress, yet there is still no tangible evidence, example, or prototype to show that life without proteins is possible. 2. The earliest unambiguous traces of life already display the presence of proteins, so there is no fossil evidence for this proposed era. As far as we can tell, life and proteins appeared simultaneously on the planet. 3. When paradigm shifts occur in biology, the preceding worlds do not simply disappear without a trace. Prokaryotes did not disappear when eukaryotes emerged. Single-celled organisms did not disappear when multicellular life forms emerged, and asexually reproducing organisms did not go extinct when sexual reproduction emerged. There is value in simplicity, especially when it comes to natural selection. So why did the RNA world disappear completely? 4. There is selective pressure that prevents the accumulation of genetic material for low-complexity life forms. This is what makes the RNA world scenario literally impossible. There is a fundamental flaw in the reasoning of evolutionary biologists. They treat information, or more precisely data, as a free resource. The prevailing theory is that genetic material gradually accumulated by successive random gene and genome duplications. But storing, preserving, and copying data is not free. It's a costly task. An organism that spends its precious resources on copying unnecessary data is at a disadvantage compared to those which copy only the essentials. This pressure is proportional to the complexity and size of the organism. It hardly affects us, but this selective pressure is crushing for a tiny prokaryote. Howard Oakman at University of Texas demonstrated in a study titled Genomes on the Shrink that every clade of bacteria with genome sizes of less than 2 megabyte was derived from ancestors with substantially larger genomes. Subsequent studies confirmed that this evolutionary pressure exists both in vitro and in vivo, affecting free-living, parasitic, and symbiotic creatures, and it strongly affects both bacteria and archaea. As it turns out, the search for finding the simplest life forms has been going on for billions of years on an unfathomable scale, and modern bacteria is the result.
Possibly the first enzyme was an RNA molecule with replicase properties.
In 2022, researchers at the University of Tokyo put this theory to the test by borrowing necessary enzymes from E. coli bacteria and acquiring the RNA template for the replication enzyme from a virus. They then began an evolutionary experiment. The original RNA template consisted of around 2,000 nucleotides. After only 200 cycles, the population was dominated by so-called parasitic sequences that were only 500 nucleotides long. This once again demonstrated the immense selective pressure for shorter nucleic acid sequences. The RNA genome shrank to a quarter of its original size in such a short time frame. This means that biological evolution could not have been started by a short piece of RNA, because the immense selective pressure to keep it short, or even shorter, would prevent the accumulation of genetic material. This force strictly prevents the buildup of longer sequences of nucleotides, demonstrating that the idea of a short piece of RNA leading to the origin of life is highly unlikely. So, to solve the problem of origins, to jumpstart life by overcoming the selective pressure for short genomes, one must give matter function, a lot of function. This is the idea behind the genome of Eden.